Let's get into episode 10 of the cell culture series. Adherent versus suspension cells, two worlds, one lab. Some cells need a surface to grow. Others thrive while freely floating in the culture medium. Although both are grown in the same laboratory, they behave very differently, and understanding those differences is essential for choosing the right cell model and using the correct culture techniques. In this episode, we'll compare adherent and suspension cells, explore their unique characteristics, learn when each is used, and understand the practical techniques required to culture them successfully. Let's begin. Some cells need to grab onto something to survive, like a climber on a rock face. Others float freely, like fish in water. This fundamental difference changes everything about how you culture, passage, scale, and harvest them. Today, the fork in cell culture strategy. Why the adhesion question changes everything. Before you design a single experiment, before you order media, before you decide on a flask size, you need to answer one question. Does your cell line grow attached to a surface, or freely in suspension? The answer determines your entire workflow, your equipment needs, your passaging strategy, and your scale-up path. The clinical relevance. Monoclonal antibody drugs, the most successful class of biopharmaceutical products in history, generating over $200 billion per year, are produced in Chinese hamster ovary, CHO, cells scaled in bioreactors. Whether CHO cells grow as adherent monolayers or in suspension directly determines whether you need a $50 trillion flask or a $2 million bioreactor. This is not a trivial distinction. Quick hook check before we move on. Why do adherent cells need a tissue culture treated, TC treated, plastic surface rather than regular plastic? Here are your options. A. Regular plastic isn't sterile enough for cell culture. B. TC treatment adds a charged, hydrophilic surface that promotes protein adsorption and integrin binding, letting cells attach and spread. C. Regular plastic dissolves in cell culture media. D. TC treatment adds nutrients to the plastic that cells can absorb. Take a second. Lock in your answer. Why do adherent cells need a tissue culture treated, TC treated, plastic surface rather than regular plastic? The correct answer is B. Tissue culture plastic is treated with plasma discharge or gamma radiation to create a hydrophilic, negatively charged surface. This promotes adsorption of serum proteins, like fibronectin, vitronectin, and collagen, which act as adhesion substrates for the cell's integrins. Without this surface modification, cells simply can't engage their adhesion machinery. No engagement means no attachment, and no attachment means no growth. Two cell worlds. Different biology, different rules. Adherent cells. The climbers. Adherent cells require a surface to grow and survive. In their native tissue, they are always anchored to extracellular matrix. In culture, they attach to TC-treated plastic via integrins connecting to adsorbed matrix proteins. They grow as monolayers until they reach 100% confluency, then contact inhibition stops growth. Now let's look at the key characteristics of adherent cell cultures. Unlike suspension cells, adherent cells grow as a monolayer attached to the surface of a culture flask. Common examples include HeLa, HEK293, A549, MCF7, and NIH3T3 cells. Most adherent cell lines originate from epithelial, fibroblast, or endothelial tissues, so attachment is a natural part of their growth. Because they stick to the flask, they cannot be collected by centrifugation alone. They first need to be detached using trypsin or, in some cases, gentle mechanical scraping. When growing these cells at a larger scale, scientists use roller bottles, cell factories, or microcarrier beads to provide more surface area for attachment. As the culture reaches 100% confluency, growth slows due to contact inhibition, so the cells must be passaged before they become overcrowded. Finally, some sensitive cell types, such as neurons and hepatocytes, require additional extracellular matrix coatings like collagen, fibronectin, or laminin to attach and grow properly. Understanding these features helps you choose the right culture conditions and maintain healthy, reproducible cell cultures. Suspension cells. The swimmers. Suspension cells grow freely in media without attachment. They are mostly hematopoietic, blood-derived, in origin. Cells that naturally circulate in the bloodstream have no need to anchor themselves. They can be passaged by simple dilution or centrifugation and are much easier to scale up. Now let's explore suspension cell cultures, which differ significantly from adherent cells. Instead of attaching to a surface, suspension cells grow freely in the culture medium. Common examples include your cat, HL60, K562, and THP1 cells. These cell lines are mostly derived from blood cells, such as lymphocytes and myeloid cells, making them ideal models for leukemia and lymphoma research. Since they don't attach to the flask, harvesting is much simpler. A quick centrifugation is usually enough to pellet the cells, no trypsin is required. Suspension cultures are also highly scalable. They can be expanded in spinner flasks or bioreactors, where growth is limited mainly by the culture volume rather than the available surface area. Unlike adherent cells, suspension cells do not experience contact inhibition. Instead, their growth is limited by nutrient depletion and waste accumulation, so regular cell counting and density monitoring are essential. Finally, contamination is often easier to spot because the culture medium becomes cloudy or turbid, providing an early visual warning. Understanding these characteristics helps you choose the right culture system and maintain healthy, reproducible suspension cell cultures. Semi-adherent, the awkward middle. Some cell lines grow as loosely adherent clusters that partially attach and partially float, HEK 293 being the classic example. They will grow in suspension under the right conditions or attached under others. Semi-adherent cells require judgment about passaging. Some labs use trypsin, some use gentle pipetting, some use both. The way you scale up a cell culture depends on whether you're working with adherent or suspension cells. For adherent cells, growth requires a surface to attach to. Scientists typically use T-flasks, roller bottles, or multi-layer cell factories to increase the available growth area. Because attachment is essential, scale-up is ultimately limited by the amount of surface area you can provide. Suspension cells are different. Since they grow freely in the culture medium, they can be expanded in shake flasks, spinner flasks, and eventually large stirred tank bioreactors that can hold thousands of liters. In simple terms, adherent cultures are limited by surface area, while suspension cultures are limited mainly by volume, making them the preferred choice for large-scale biopharmaceutical manufacturing. Now it's time for another core concepts check. Here's your question. You need to produce 500 milligrams of a monoclonal antibody from CHO cells. You can either scale up using multiple T175 flasks or a stirred bioreactor. Which option produces more protein, and why? A. T175 flasks because they provide better gas exchange per cell. B. A bioreactor, because suspension-adapted CHO cells scale by volume rather than surface area. C. Both produce the same amount, it's only a cost difference. T175 flasks because adherent cells naturally produce more protein. The correct answer is B. Here's why. This is exactly why the biopharmaceutical industry relies on suspension bioreactors for manufacturing therapeutic antibodies. With adherent cells, production is limited by the available surface area. Increasing output means adding more flasks, more incubators, and much more manual work. In contrast, suspension-adapted CHO cells grow throughout the entire volume of a bioreactor, allowing production to scale efficiently. In fact, a single 10-liter bioreactor can replace more than 200 T175 flasks. That's what makes large-scale production of biologic drugs both practical and economically viable. Passaging adherent versus suspension, two different protocols. Adherent cell passaging, HeLa example, 1. Visualize at microscope, confirm 70 to 80% confluency, healthy morphology. 2. Aspirate spent media completely. 3. Wash X2 with PBS, removes serum trypsin inhibitors. 4. Add trypsin EDTA, 2 ml per T25, 3 ml per T75. 5. Incubate 3 to 5 minutes at 37 degrees Celsius. Monitor for cell rounding and detachment. 6. Neutralize immediately with complete media, 2 to 3x trypsin volume. 7. Count viable cells, calculate split ratio, seed new flasks. Suspension cell passaging, your cat example. 8. Check cell density by hemocytometer or OD. No microscope adherence check needed. 9. Transfer entire suspension to 15 ml conical tube. 10. Centrifuge 300 grams by 5 minutes. 11. Aspirate supernatant. Resuspend in fresh complete media. 12. Count viable cells. Seed new flasks at 2 to 5 by 10 to the power of 5 cells per milliliter. No trypsin, no PBS wash, no waiting for detachment. The entire process takes 10 minutes versus 20 to 30 minutes for adherent cells. The trade-off: you must monitor cell density carefully because there is no visual confluency cue. Density monitoring for suspension cells. Suspension cells do not give you the visual confluency signal that adherent cells do. You must count or estimate density at regular intervals. Letting suspension cells overgrow, greater than 2 to 3 times 10 to the 6 per milliliter for most lines, leads to nutrient depletion, pH crash, accumulation of lactate and ammonia, and rapid culture collapse. Set a regular monitoring schedule and stick to it. Now, let's test your understanding with a quick lab demo quiz. Here's your question. You accidentally add trypsin to your cat suspension cells, thinking they were HeLa cells. What is the most likely consequence? A. No consequence. Trypsin does not affect suspension cells. B. Your cat cells will rapidly attach to the flask and become adherent. C. Trypsin will cleave surface proteins on your cat cells, affecting their biology, viability, and any surface marker expression you plan to measure. D. Trypsin will cause your cat cells to differentiate. The correct answer is C. Here's why. Trypsin doesn't distinguish between adherent and suspension cells. Its job is to digest cell surface proteins, regardless of the cell type. In your cat cells, trypsin can remove important CD markers, cytokine receptors, and other surface proteins. This can affect cell function and produce inaccurate results, especially if you're planning flow cytometry or any experiment that depends on intact surface markers. That's why suspension cells should never be trypsinized. Instead, they should be collected by centrifugation and resuspended in fresh medium. 3 mistakes in the $200 billion industry. Mistake 1. Using non-TC treated plastics for adherent cells. The fix: tissue culture treated flasks, dishes, and plates have TC in their product name. Regular plastic does not. Adherent cells in untreated plastic will not attach or will attach poorly. Always verify TC treatment status of every vessel before use. Mistake 2. Not monitoring suspension cell density. The fix: set a regular counting schedule, every 24 to 48 hours for fast-growing lines. Passage when density reaches 1 to 2 times 10 to the 6 per milliliter, line dependent. Do not rely on visual turbidity assessment alone. By the time turbidity is obvious, the culture may already be crashing. Mistake 3. Trypsinizing suspension cells. The fix: suspension cells need only centrifugation and dilution. If you find yourself reaching for the trypsin for a suspension culture, stop and reconsider. The only exception: if suspension cells have accidentally adhered, some semi-adherent lines, gentle trypsinization may be needed to detach them. Now it's time for the final boss quiz. Here's your question. You are designing a process to produce 1 kilogram of a recombinant therapeutic protein using HEK 293 cells. What is the fundamental scale-up limitation of adherent HEK 293 cells, and how can it be overcome? A. Adherent HEK 293 cells cannot produce recombinant proteins. You must use E. coli. B. Adherent cells are surface area limited, so large-scale production requires either exponentially more flasks or adapting the cells to suspension culture in bioreactors. C. HEK 293 cells cannot survive in cultures larger than 1 liter. D. There is no fundamental limitation. Adherent and suspension cultures scale the same way. The correct answer is B. Here's why. One of the biggest concepts in bioprocess engineering is understanding how different cell cultures scale. Adherent cells grow only on a surface, so increasing protein production means providing more surface area, more flasks, more cell factories, and more incubator space. Suspension-adapted HEK 293 cells, on the other hand, grow throughout the culture medium. This allows production to scale by increasing the culture volume in bioreactors, making large-scale manufacturing far more efficient. That's why most industrial HEK 293 production today uses suspension-adapted cell lines to overcome the limitations of adherent cell culture. Episode 10 Summary. What you now know. 1. Adherent cells need surfaces, suspension cells float freely. This fundamental difference in biology drives completely different culture strategies, passaging protocols, and scale-up approaches. Know which type you are working with before you design anything. Putting suspension cells in a tea flask and hoping they attach is not a strategy. 2 Each type requires different passaging, monitoring, and scale-up strategies. Adherent. Visual confluency check, trypsin, density calculation. Suspension. Count-based monitoring, centrifugation, dilution. Mixing up these protocols leads to damaged cells, trypsin on suspension, unattached cells, no TC treatment, or culture crash, ignoring suspension density. 3 Blood-derived cells are almost always suspension, tissue-derived are usually adherent. This heuristic gets you to the right protocol 90% of the time. HeLa, cervical, MCF7, breast, A549, lung, equals adherent. Your cat, T-cell leukemia, HL60, myeloid, K562, CML, equals suspension. When in doubt, check the ATCC datasheet. Thank you for watching episode 10 of Cell Culture series. In the next episode we'll dive deeper into transfection. See you there. If you found this episode useful, don't forget to subscribe our channel. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.